Salt Run is a 3-mile-long tributary of the Matanzas River estuary, located in northeast Florida. Previous modeling studies performed by Tricia Kaiser classified it as a priority focus water body due to abundant septic systems and elevated nitrogen discharge. The objective of this research was to examine the spatial and temporal variation in water quality parameters at 5 sites along the length of Salt Run. We hypothesized that as we progress further south into Salt Run, away from the mouth of the inlet, levels of ammonium, BOD, Enterococcus, and fecal coliform will increase. By completing this study, we can contribute to baseline data, identify any harmful levels of bacteria, protect the Salt Run marine ecosystem, and conserve the health of the community.
Once a week for 7 weeks, the team loaded up with St. Augustine Eco Tours and went out to Salt Run for sampling. We worked backwards, starting at Site 5 to make our way forwards to Site 1 at the mouth of the inlet. At each site, we used a YSI multimeter and DO probe to measure salinity, pH, dissolved oxygen, and water temperature. We used the depth finder to record the depth at each site. At sites 1 and 5, we collected 4 water samples to be used to measure BOD and stored all the water samples in the dirt cooler. We collected duplicate samples at each site by using a 3-meter-long pipe with a stopper on the end to collect samples of the entire water column. The water was emptied into a bucket, and using a pitcher, we rinsed each bottle 3 times before filling it and storing it on ice.
After measuring the initial dissolved oxygen, the bottles were placed in a dark bucket on a shaker for a 5-day incubation period. Final dissolved oxygen concentrations were then measured to be able to calculate BOD. Enterococcus was measured for sites 1, 3, and 5 using the IDEX Quantitray method. 10 milliliters of unfiltered sample, 90 milliliters of deionized water, and 1 capsule of enterolite reagent were combined. The mixture was shaken and then poured into a tray. Each tray was sealed using the IDEX sealer and then placed in a water bath of 41 degrees Celsius to incubate. After 24 hours, the trays were placed under a UV light in order to count the number of cells lit up with Enterococcus bacteria. Fecal coliform was measured at all 5 sites for 3 sampling dates and analyzed following the EPA membrane filtration method. 100 milliliters of sample was run through a gridded filter. A sample dish was treated with fecal broth. The filter was transported to the sample dish, sealed, labeled, and placed in the oven at 44.5 degrees Celsius. After 24 hours, the sample dishes were removed from the oven and the number of fecal coliform colonies were counted and recorded. Ammonium was analyzed using the method of Holmes et al. 1999. 250 milliliters of each sample was filtered and poured into an acid-washed bottle. 20 milliliters of filtered sample was combined with 10 milliliters of an OPA borate buffer solution. Background fluorescence was taken using a Trilogy Turner fluorometer. After 2 to 3 hours of incubation in the dark, the final fluorescence values were taken. All of this data was recorded and manipulated using Excel.